In this video, I'm going to explain about transcription and translation, which is the process of protein synthesis. Before we begin, however, we need to look at the structure of RNA, because RNA plays a crucial part in protein synthesis. RNA is another nucleic acid, just like DNA, and so it is made up of nucleotides. The phosphate is exactly the same. However, the rest of it is ever so slightly different from DNA. First of all, the pentose sugar is ribose rather than deoxyribose, and the nitrogenous bases have uracil instead of thymine. This means that A now pairs with U rather than with T. When we're looking at protein synthesis, we have a bit of a problem because the DNA, which contains the genetic code, is in the nucleus, whereas the ribosomes, which is where protein synthesis takes place, are in the cytoplasm. So what we need to have a messenger, and that messenger takes the form of RNA, specifically messenger RNA. Now, messenger RNA is single-stranded. So if we look back at our picture of the RNA nucleotide, messenger RNA is very straightforward. It is simply a single-stranded molecule which can be made to copy DNA. So what happens in the first stage is we need to create this messenger. So DNA in the nucleus unwinds and unzips in the region of the gene to form single-stranded sections here in what we call the replication bubble. Now, in DNA replication, it was helicase that did this, but in transcription, it is a different enzyme called RNA polymerase. So RNA polymerase unwinds and unzips the DNA, and it also brings in the complementary RNA nucleotides so that they form a temporary bond with the DNA. Now, they form a temporary bond with the DNA on what we call the antisense strand, and so the antisense strand is complementary to the mRNA code, which means that the sense strand, which is the strand which the RNA does not attach to, is the same. So we say that the sense strand is the coding strand because that carries the same code that the mRNA has. So the RNA polymerase will work along, chug, 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 bringing in the complementary RNA nucleotides, and then when it gets to the end of the gene, it reaches a terminator and it will sort of fall off. At this point, the mRNA will detach from the DNA, and the DNA will then wind itself back up, and the mRNA will move into the cytoplasm, where it will then attach to a ribosome. And that is transcription. The next stage is translation, and in translation we need to be introduced to another form of RNA. So again, it's going to be made up from the same RNA nucleotides, but this RNA we call tRNA or transfer RNA, and it's structurally slightly different from mRNA. It has double-stranded sections and it has single-stranded sections. So these sections here are double-stranded, but then at the ends of the arms we have these single-stranded loops. Now, this loop is particularly important, and it's got 3 nucleotides, and we call it the anti-codon loop. And it is those 3 nucleotides are really, really important, those 3 bases, because they then control the next phase. So going back to our mRNA, so our mRNA is now attached in the ribosome, and you can see in this ribosome that we've got these little clefts, and they are exactly 3 codons wide. So we read the genetic code in triplets, because if we read it in triplets, then that gives us enough possible combinations to make 20 amino acids. Remember, if we read it in pairs, we would only have 16. If we read it singly, we would only have 4, and we need to have 20. Now that makes the genetic code degenerate, which means that there are more codons than there are amino acids, but that is useful in itself. So the first stage of translation is what we call amino acid activation, and this involves the tRNA molecules and it involves the amino acids. So the tRNA molecules attach to an amino acid. Now this is super clever because the 3 bases on the anticodon loop determine which amino acid joins in there. The complementary amino acid to the codon here, so the complementary anticodon, will then come into the ribosome like this and bring into it its amino acid. Then in the next cleft, exactly the same thing will happen. So the tRNA with the complementary anticodon loop to the codon exposed in the ribosome will bring in its amino acid. These amino acids are now close enough together for a peptide bond to form between them, and then the ribosome will move on, and so on and so on and so on, and in doing that you end up with a complete protein.